It's great being the first speaker here. Partly what I'm doing now with my talk here will be to introduce some of the work of the next 5 speakers who are all working on projects that I'm associated with. But I also wanted to go through a range of projects that I've been doing. And then if there's time, I'm going to go into some actual data. So I have worked on a number of ontology projects for many years. So these include the cell ontology and the gene ontology. Recently in the cell ontology, I've gotten more active in collaboration with David Sutherland, whose group in England at the Sanger Institute has really taken over the lead in development of the cell ontology in the last 4 or 5 years. And they're using a lot of AI methods now to develop ontology definitions. And they've implemented Chris Mungel's AI-driven approach to ontology editing, where you make a request in a GitHub issue, and there's agentic AI that goes out and will change the ontology. This is active for both the Go and the cell ontology and several other ontologies for Uberon, for instance. And basically, the agentic AI implements the changes and then creates a PR, and then a human can review the PR. And so I'll be doing some of those with them, particularly as it relates to immunology, immune cell types. I'm also in a very big collaboration. Well, the collaboration is very big. It's among quite a number of people who work in a group called SOLCAP, which is organized by Ryan Brinkman. And these are international collaborations of researchers in flow cytometry who work in academia and industry. And they're providing definitive flow cytometry-based definitions of immune cell types. And my eventual job with them, which I'm still kind of waiting on their definitions to be finished, will be to compare those definitions to what we already have in the cell ontology and perhaps improve the cell ontology definitions or add new cell types if necessary. And David Sutherland has also helped me already by starting an agentic workflow to help me do that comparison. I've also been reviewing assignments of cell types to single-cell RNA expression data for immune cell types. And I'll be continuing that work, looking at data from HCA and HubMap in the future. I still consider myself somewhat engaged with the gene ontology, although mostly it's responding to particular issues from time to time that I think need my input. So my group a few years ago developed this protein conformation ontology, which represents secondary, tertiary, and quaternary aspects of protein structure, actually as dispositions. And we have a paper that we wrote. It was initially rejected, but we're going to revise it and improve the ontology a bit. And the people who've been most involved recently have been Giacomo, who's, of course, a philosophy graduate student, although not actually present today. I've also been involved with the Kidney Precision Medicine Project, looking at single-cell RNA sequencing data in CKD and AKI. I had a summer student last year who continued to work with me, Eddie Munez. And this work has been done in collaboration with Oliver He and his group is also doing similar analyses, and I think it'll be quite interesting to compare the two approaches. As some of you know, I've worked on a number of ontologies related to neurological disease. These are mostly inactive now, but what's interesting is the neurological disease ontology itself. The paper that was published in 2013 often comes up as one of my top 5 most read papers on ResearchGate, even this year. So there seems to be continuing interest in the topic of neurological diseases and ontologies for them. The neuropsychological testing ontology And both these ontologies were developed largely by Mark Jensen and Alex Cox. NBT, I think it's actually quite a good ontology, but been waiting for the right student for a number of years to take over the work that Alex Cox started. And then there's multiple sclerosis patient data ontology. This was originally work of Mark Jensen, but then Mackenzie Smith, who was an undergraduate at the time, along with my graduate student, Lauren Wishney, we completed this large analysis of MS patient-reported outcomes data, which at some point I should make the subject of another talk. Basically, this data includes data about disability as well as about patient afferent responses or emotional responses to the disease, and it's from patients over the continuum of the disease. It's quite interesting data. I'm in a very current project called the Neuroelectronic Interface Ontology. So current that Bill and I are part of a grant that's being submitted today and that we were working on last night even. This project represents bionic vision devices and the diseases and conditions under which they can be used. So these are devices that can be implanted in the head or near the head, and they can interpret visual data to be fed into the neurological system. And there's been quite a lot of work on this. And we're working with Daniel Ratburn of Henry Ford Health and Michael Byler of UC Santa Barbara, who are experts in this field. And actually the work here is really nice because it builds on earlier work that Patrick Ray did with me when he was a graduate student on building something we called the ocular disease ontology, and also work that Patrick did that provided an ontological representation of vision and blindness. And so we're going to be able to create a phenotyping algorithm to look at EHR records that will be based in part by using this ODO ontology and also an ontology to be built based on Patrick's work that will describe visual function in various ways. So directly after me, Alex Bartnick will be talking about the magnetic resonance imaging acquisition and analysis ontology, which represents methods and data types for magnetic resonance image acquisition analysis, including fMRI. And recently we've added a lot of statistical methods for imaging. Many of the upper level terms here have been exported or gifted to the Ontology for Biomedical Investigations, and we have a whole set of new terms that we want to be giving them soon. The point of putting them in OB is that it makes them more readily available to a wider range of people working in the biomedical ontology area. And this work has been done by Alex Bartnick, started by my earlier student, Lucas Serra. Alex Terry was a summer student last summer who worked on the fMRI stuff, and Lauren Wishney and Bill Duncan have also contributed. And Alex Bartnick and Bill Duncan and I are part of this project to build an imaging ontology for biomedical applications, which is incorporating MRI into a more general representation of biomedical imaging. And recently, Sam Border has started work on doing a representation of pathology imaging methods that will fill in that gap in what MRI doesn't do. And we had previously applied for money with a guy named Carl Helmer from Massachusetts General Hospital, which was not successful, but we'll probably submit that again, particularly, I think, after Harvard gets out of purgatory. This is actually basically another OB development project overall, because many of these terms will be transferred to OB once they're ready. So my group is, or I'm part of a group working on ontologies related to, let's put it as pain and teeth. So there's the pain ontology, which Finn Wilson is going to talk about later, I believe. And this covers types and mechanisms of pain And we have a collaboration that includes both Roger Filligum from Florida, who's a pain expert, and Bronwyn Freeman, who's also a pain expert from Germany. Also, I've had a student, Ava Cunningham, who's going to talk later, who's been working on a conversion of what's called the International Classification of Oral Facial Pain, which is a terminology about oral facial pain. She's been converting it into an ontology, meaning she's adding axioms and updating definitions and so on. And this work is actually part of the larger oral health and disease ontology, which Bill Duncan and Alan Ruttenberg and others worked on in the past and are continuing, and Bill's continuing to work on it now. And Finn has also contributed to this work. And then there's the ontology of dental care related fear and anxiety. So this is a grant-funded project. Bill and Dan McNeil are the lead in Florida, but I'm a co-investigator on this project. This represents types and conditions of dental fear and anxiety. It represents survey data to enable secondary analysis of that data. It is also part of the oral health and disease ontology. And the very subset that represents ODFA has been separated and, wow, moved to BioPortal. And we have a lot of collaborators. Continuing work on immunology, ontology related things. So we've had a long series of discussions of immunology and developing an immunology ontology. At one point, I did collect a whole set of terms from Go and cell ontology with the help of James Overton. A lot of folks are interested in this project. What's happening more recently is that Aaron Abend, who runs this autoimmune registry, we're planning to do a grant submission now to develop an autoimmune ontology, which would really be part of a larger immunology ontology. And that would be done with Anna-Maria Meshi and Asiya. And I currently have an undergrad, Chris Thacker, who's doing some LLM work to extract data about autoimmune disease from a textbook. Anna-Maria is also interested in developing a tumor microenvironment ontology. And years ago, I worked on this antibody ontology. That might actually be revived as part of this SOLCAT project, which I discussed earlier. Cancer ontology work. Bill and I worked on this neoplasm ontology some years ago. And then I had a summer student who added classes related to ovarian cancer. And I have a new student who's going to continue the work on ovarian cancer. A separate project is the cancer cell ontology, which my student Lucas Serra did a couple of years ago, which was basically representing leukemias, lymphomas, and myelomas based on a standard from Europe called EAGLE, which is a flow cytometry standard. It was based on the same approach as the cell ontology, uses the cell ontology as an upper level work. And actually, this work may also be revived as part of the SOLCAT project because they want to cover certain types of leukemias as well. And then the cholangiocarcinoma ontology was done by my graduate student, Anawat Pengput. It represents cholangiocarcinoma and related entities. And Anawat, for his dissertation work, among other things, did an analysis of risk factors in the development of CCA, which is an endemic disease in Thailand because of people being infected with liver flukes they get from eating infected fish. Anawat also worked with Werner Koisters to convert this ontology to a first-order logic format, which is really interesting work, but separate from my work with Anawat, except for the fact that they used the work that we did together as a basis for that development. So you're saying I'm out of time, so this is a good time to stop.
Very good. Thank you, Alex.